This is Zhe Yu from University of Ottawa. Our topics of poster is gaps and runs in syntenic alignments. Gene loss is obvious of novel gene acquisition by a genome through process such as whole genome duplication. During gene loss events, some of the copies of a gene will be deleted by functional or structural mechanisms. This process suffers energetic and material cost of gene complement expansion. We use the data of L-tetraploid coffee arabica and its two diploid progenitors, coffee canaveral and coffee eugenioides. Here's the circles graph where we can see the relationship between the syntenic box in subgenomes of coffee arabica and its partner genomes canaveral and eugenioides. The evolution history of these polyploids are shown here. There are three main genomic events: gamma whole genome tripling, CCC speciation, and L-tetraploidization. Then we use the peak method to locate the three events in paralogous and orthologous syntenic blocks in 10 self and pairwise comparisons between subgenomes and genomes. In this poster, we mainly focus on the frequency distribution of gap lines within syntenic blocks. First, we set a simple model with the assumption that one gene is randomly selected and deleted at a time. We call it one-at-a-time model. Starting with an array of ones with length n, at each step, we locate a 1 by random variable from discrete uniform distribution with current length, and then transfer this 1 to 0. We use mtx as a notation for the expected number of runs of ones of length x at time t. This is a proved equation for recurrence of mtx. This equation explains how we gain and lost the run length. mt minus 1x represents the number of run lengths x at previous time. mt minus 1x over the current total length gives the probability that new deletion located at some run length x. X times the probability returns the expected value of lost runs. Same thing for the third term. Twice the probability that new deletion located at any longer runs shows the expected value of creating new one from either two sides of the runs. In the diagram, we compare the simulation and the real data. Colored lines represent the number of runs of lost genes after speciation and after tetraploidization. And the black dot line represents the simulation of one-at-a-time model. Each line represents a typical stop time. We can notice that the real data is syntenic departure from this model. No matter how we change the total length or the stop time, The model is inadequate to account for both the simultaneous steep drop-off from gap size 1 and the presence of significant long gaps. Because the poor fit of this model, we try to combine this model with gamma distribution, and then optimize the gamma parameter by chi-square criterion. The optimized parameter of gamma turns out to be a geometric distribution, and this returns a 70% of dilation from one at a time model and the rest from geometric distribution. And from the diagram, we can see the combined model is much better fit at both the head and tails with the real data. Thank you for watching.